Hello, welcome back to the course on Polymeric Biomaterials. So far, over the last few lectures, we have been focusing on the theme of bulk behavior and responses, as part of which we have largely been looking at physical and mechanical behavior and responses, namely mechanical testing, viscoelastic responses, and so on. In this lecture, we will continue that theme, but we will switch gears a bit and now start to look at degradation of polymeric biomaterials, where we will revisit some ideas from our earlier lectures about backbone chemistry, side chains, crosslinks, etc., and see how all of that can play a role in determining the degradation of polymeric biomaterials, which is a very important topic when considering biomaterial design. So, as part of today's lecture, we will be looking first at a few terms that are commonly used in the context of degradation. One is degradation and or biodegradation, the other is erosion, and the third is resorption. And our goal is to see if we can understand the differences between these and try to contextualize them. We will also look very briefly at two other words that we have looked at in quite some detail in earlier lectures and see if those have anything to do at all with the topic of degradation. And then we will move on to understanding how crystallinity, stereoregularity, molar mass, essentially polymer properties, the properties of polymeric biomaterials, how they impact degradation. And then we will look at some mechanisms of degradation, broadly along two categories, physical breakdown and chemical degradation. And then we will also look at some specific mechanisms such as oxidative, hydrolytic and enzymatic. And then towards the end, we will loop it back to our idea of biomaterial design and ask how do we understand or contextualize degradation against the backdrop of biomaterial design for specific applications. Essentially, what factors should we consider for biomaterial design, especially when you look at degradation. So, with that quick introduction, we will jump right into today's lecture. And the first thing we will do before we actually look at degradation is spend a little bit of time appreciating the importance of the aqueous microenvironment that all biomaterials will be subject to when they are implanted. So, the buffer in the body is an aqueous buffer with salts, and largely what we will be looking at in terms of degradation will relate to how water can interact with the biomaterial, enter the biomaterial matrix and then start doing things that can eventually lead to degradation. So, it is good for us to look at some aspects of the aqueous microenvironment, particularly properties of water and how we can contextualize it for degradation. Water is something we take for granted. It is very underappreciated in terms of its properties. Water is one of those solvents that can extensively form hydrogen bonds, both intra and inter, and life in some sense, right, was possible only because the solid state ice has lesser density than liquid water, right. Aquatic life would not have been possible if this wasn't true. So, and this is unique to water. Also, given its molar mass, very small molar mass, water has extremely high freezing and boiling points. Just as a test case, if you take hydrogen sulfide, right, where the only change we have made is to go from oxygen to sulfur, now you are talking about a gas essentially at STP, normal temperature and pressure, and you know, if you want to look at freezing and boiling points, you have to go to -80 and -60 kind of temperatures, degrees Celsius, for hydrogen sulfide. Now, compared to that, you see where water is, right, much higher freezing points and boiling points, essentially a liquid at physiological temperatures, and that is sort of what we are saying here. And water also has other very unique properties. It has an extremely high surface tension and specific heat, and in some sense it's called the universal solvent because all the biomacromolecules that we have seen in this course exist in the aqueous medium of water. Proteins, polysaccharides, even lipid bilayers, ultimately they have to exist in an aqueous microenvironment. They will find a way to self-assemble, no doubt, but they still have to exist in an aqueous microenvironment. Ions, many of the ions that are responsible for regular physiological function, gases, all of these have to exist in the medium of water. So, water is an extremely important but very underappreciated aspect of biomaterials, because ultimately it's the interaction of water with biomaterials that will ultimately determine the initial and eventual performance of biomaterials. Now, that leads us to this idea of biomaterials in an aqueous microenvironment. And if you just think about how would you model water, right? How would you think of the interactions between water and the biomaterial? And if you have to think about it from a modeling perspective, for example, from a molecular dynamics perspective, right? You will find that there are several different models and force fields available for water, right? Because no one force field or model can capture all properties of water. So, the model that or the force field that you use for water will depend upon what properties are of interest in your specific modeling, right? Of course, the presence of ions, dissolved solutes, etc. is going to alter the structure, right? And just as an example, we have seen when we looked at hydrogels, we saw that water, you can at least think about two different forms of it, both free and the bound water, right? Free, which is the bulk, and the bound water, which is in close association with the polymeric chains. And that is going to influence things like diffusion of biomolecules, how cells attach to biomaterials, all of these are going to be influenced by the nature of water that we are talking about, right? And once you implant materials eventually, or you put them in culture, in cell culture, the first thing that we have sort of hinted at is going to happen in earlier lectures is that proteins are going to come and adsorb, right, on the surface. But before proteins can come and adsorb, the material surfaces usually first see the water and then proteins come and adsorb, right. So, any protein adsorption models that are meaningful will account for the presence of surface water, right. Now, protein adsorption is something we will look at in a future lecture when we talk about surface properties, but remember here our goal is to look at all of this in the context of bulk properties, particularly what water can do in terms of both plasticization and degradation, right. Plasticization is something, again, we have seen water in some sense is a universal plasticizer, right. Once it gets in, once it starts interacting with the biomaterial, it can increase the free volume, lower the Tg, essentially that is what a plasticizer is, right. And that can have a large impact on the mechanical properties also, right, on the performance of the biomaterial. And in this lecture, our focus is mostly what water is going to do to degrade the material itself. So, put a pin on this, we will be revisiting this idea of interaction of water and how that eventually dictates how a biomaterial will degrade in the microenvironment throughout this lecture. So, before we proceed to look at degradation mechanisms and what the effects of polymer properties are, we will pause for a bit and look at a few terms that are commonly used in the context of degradation. This could be a point of confusion for many, so it is good to clarify some of these things. First is the word degradation itself, and essentially it refers to a chemical process by which bonds are cleaved. So, when we talk about degradation here, we are referring to two things. One is decrease in the molar mass and a loss of properties, most notably mechanical properties. Right. So, when we say decrease in the molar mass, you immediately have to be able to relate it to cleavage of the backbone itself, right. If molar mass can be correlated to chain length, then a decrease in molar mass should indicate to you that the chain length is going down. And one way for that to happen is if the covalent bonds in the backbone itself can now be snipped off, right? So that's what we're talking about here. And one example of that is PLGA hydrolysis, right? PLGA is a copolymer that we have looked at in many lectures as part of this course, and it's a polyalpha ester, and it can undergo ester hydrolysis. Depending on the lactate to glycolate ratio, the rates can be tuned, rates of degradation can be tuned, and we will look at that later in this lecture as well. And for degradation, what matters is the local microenvironment is quite critical. One example of that is just pH, right? pH can have a very significant role. Localized changes to pH, or if the environment itself is acidic, such as the stomach, then that's going to have a very different effect on degradation compared to the rest of the body, right? So that has to be contextualized. Now the other word that you will often see is biodegradation. Essentially, it's the same as degradation, where you're still cleaving bonds or bond scission occurs along the backbone, except now this is happening due to a biological agent such as an enzyme, right? And shown here is the example of gelatin, which is simply, as we have seen earlier, a denatured form of collagen, right? And gelatin can be degraded using specific enzymes that are found in the body. For example, one such enzyme or class of enzymes are called matrix metalloproteinases or MMPs. So there are a class of enzymes that can degrade proteins, and gelatin being a protein can be enzymatically degraded by MMPs. The other aspect to consider apart from degradation and biodegradation is this idea of erosion, right? Now in many contexts this distinction is not made, and the distinction between degradation and erosion remains quite fuzzy, but they are actually not the same. Erosion actually is a process that results in mass loss of the material. So once degradation starts occurring, erosion will continue, because ultimately erosion is going to lead to changes in physical size and shape of the material itself in the processed form, and this can occur either in the bulk or via surface driven mechanisms, depending on the interaction of water with the material and what type of bonds are present within the material, right? Now just as an example, again we are looking at PLGA, which would be considered bulk degradation, and polyanhydride erosion, which would be considered surface erosion. Now ultimately the end result of erosion is what you would see in the biomaterial is gross macroscopic changes to physical size and shape, right? The third word we are going to look at is resorption, right? We talked about resorbable sutures earlier. Now resorption is, again, these are all related, but there's some subtle differences between the words. Resorption has to do with the processes that lead to elimination of the degradation products. So once the material has degraded, the process of eliminating the degradation products is generally what is called resorption. And PGA, for example, polyglycolic acid, can resorb through several metabolic pathways. And as far as resorption is concerned, one thing to keep in mind from a device perspective when using biomaterials as part of devices that can be implanted in the body is the nature of end products of degradation need to be carefully considered for safe elimination from the body. So not only are you looking at efficacy of the material in doing its performance, you are also looking at safety. Again, these are the two pillars upon which any standard approval agency such as the FDA is going to look at your material or device, right? So that's degradation, erosion and resorption, and we will look at some examples of each of these things throughout this lecture for us to get a better grasp of the concepts such as erosion, degradation and resorption. Now two other words that we have seen and you would often again come across are dissolution and aging, right? And one question you can ask is, are they related to degradation? Are they the same as degradation, etc.? The short answer is they are not the same, but they could occur along with degradation. For example, Dissolution is simply you taking a polymer, and this is something we have seen in lecture 14 in great detail, where you take a polymer and you dissolve it in a solvent, right, where we have seen the different stages of solvent entrainment into the matrix, swelling, gel formation, and then eventual dissolution, where primarily you are talking about a reduction in or the breaking up of secondary interactions, right, and eventual solubilization. Whereas in degradation, remember, we are talking about a decrease in molar mass, where you are breaking apart the covalent bonds in the backbone. Those are not secondary interactions, right. So, clearly you see dissolution is quite different from degradation. Likewise, aging is another concept we saw in detail in lecture number 23. And while aging can occur along with degradation, aging is quite different. Physical aging, for example, can include mechanisms such as molecular relaxation. Once the material is implanted, once it starts interacting with water, once it's at 37 degrees Celsius, depending on the TG of the polymer, you can have molecular relaxation happening, you can have localized crystallization happening, you can have some kind of phase separation happening. So, you see, these also lead to changes in localized changes, perhaps eventually leading to bulk changes in material properties, but that is not the same as a decrease in molar mass that you will see with degradation. So, while they are can be related and aging can occur along with degradation, they are not quite the same. And I think that is important for us to stop and pause and reflect on these ideas before we proceed further. So, now that we have sort of broadly defined what degradation, erosion, biodegradation and resorption are, and we have distinguished it from aging and dissolution, we will now quickly look at the polymer properties. From a polymer perspective, what properties govern degradation, and how can we understand biomaterial design from a polymer structure property perspective, such that we can now tune or tweak these properties to eventually control degradation, right. The first thing you can look at is the backbone functionality itself, right, because we said degradation has to do with scission or cleavage in the backbone itself. That is one aspect of degradation that is very important, leading to a decrease in molar mass. So, if you have groups, functional groups in the backbone that are susceptible to hydrolysis, now that can undergo, the polymer can undergo degradation. Polyanhydrides, for example, as we have seen, can be used for short-term drug release. They are largely going to undergo surface erosion kind of a degradation mechanism. Polyalpha esters such as PGA, PCl, etc., with tunable degradation rates, they can undergo bulk ester hydrolysis, right. Again, depending on the type of polyalpha ester you are talking about, for PGA you can have rapidly degrading sutures, because PGA is going to degrade quite rapidly, as compared to PCl, because PCl degrades much more slowly. You can use it for implantable long-term drug release kind of applications, right. So, you see, again, we are connecting the rate of degradation of the material to its eventual application. So, we are connecting in some sense a property that ultimately dictates how the material is used in a particular application. That is the connection we are making here, right. Now, in terms of functional groups, if you had to look at a spectrum of functional groups and you put them on a timeline such as this, and you think of the relative hydrolytic rates of the different functional groups, on the one hand here you have these polyanhydride functional groups that will degrade very rapidly. You see, the T half is of the order of a few hours, but again depends on the polyanhydride. But typically for the applications we are talking about, such as the gliadal wafers, where you are going to use poly CPP, sebacic acid 2080, it is going to degrade over the course of a few days to a couple of weeks, right. Now, on the other hand, you have something like polyamide, right. Nylon, for example, is a polyamide. It is going to take much longer to degrade, right, because polyamides are not that easily susceptible. Again, they can undergo hydrolysis eventually, because remember you are looking at hydrolytic rates of functional groups on a relative basis, but this probably explains why nylon can persist for very long, right? When nylon sutures are used, they are for all practical purposes considered non-degradable, right? And somewhere in between, you can have something like a polyester, right? Like PGA, PCL, etc. And even here, there is some subtlety. So what must be kept in mind is the functional group is just one aspect of degradation. For example, if you take polyethylene terephthalate, right, which is also a polyester, it's not going to degrade quite rapidly. It's going to degrade quite slowly. Can you, from our understanding and discussions on structure property relationships earlier, try to understand and speculate why PET does not degrade rapidly? So if you've paid careful attention to the structure property relationship lectures, you will be able to immediately recall that the polyethylene terephthalate in the backbone has hydrophobic bulky linkages, right? So water is not going to go in as easily, both because it is hydrophobic and because it is semi-crystalline, right? So you see, just saying that polyester has intermediate rates of degradation compared to polyanhydrous or polyamides is sort of a gross generalization. There's a lot of subtlety that has to be kept in mind because functional groups are only one aspect of it. They are important, no doubt, but they're only one aspect of degradation. There are other aspects such as hydrophobicity and crystallinity that also play a very important role in how water can enter and cause hydrolysis. So that something PET is one example of such a polymer that although it's a polyester, it's going to take much longer to degrade. Now, if you think about other polymer properties such as molar mass, right? Higher the molar mass, longer your chains are going to be and higher the interchain forces, right? And like we've said continuously in this lecture, water has to enter first and break apart the secondary interactions and then cause hydrolysis along the backbone. So if you have very long chains with extensive secondary forces, interchain forces, etc., access to the backbone for water and enzymes is restricted, which means typically larger the molar mass, slower is the degradation. So we already see that at least there are two ways to control the degradation and degradation rates of polymeric biomaterials. One is by simply looking at the backbone functionality, and even here, for example, we saw that if you only have the CPP, it's going to take a few years to degrade. But now the introduction of the sebacic acid as a copolymer in the backbone now brings down that degradation rate from a few years to a couple of weeks, right? So just by changing the functionality in the backbone, I can change the degradation rates. And again, we have seen that that can be of very great relevance in these gliadal wafers where this drug carmazine, this is an anti-cancer drug, is loaded and for glioma patients before the after the tumor is removed, before the head is sutured up, these wafers are placed inside and they release this drug over time, right? The other way to control degradation rates is by controlling the molar mass, right? So these are at least two aspects you have seen. There are some more as we will look at on the slide. Now moving on to looking at architecture, microstructure and hydrophobicity. So we have seen in lecture 10 the importance of polymer architecture. We spent quite a bit of time understanding linear versus branched versus cross-linked. And again, what I want you to recall at this point is the interactions that govern the structures between linear, branched and cross-links. Once you've been able to understand and recall what kind of interactions can exist in these kinds of structures, then it will be quite easy for you to understand that branched polymers are going to degrade faster in general, right? Branching typically increases degradation rates. If you have cross-linked polymers, for example, elastomers, right, or thermosets, highly cross-linked, they are going to resist degradation because those are covalent cross-links, right? So once formed, it's not going to be easy for thermosets to get degraded at all. Whereas in hydrogels, for example, you can have cross-links there But again, remember, crosslinks in hydrogels, if you are clever enough, you can engineer degradability into the crosslinks, right? So, into the crosslink itself. So, if you make the crosslinks, right, susceptible to certain kinds of degradation mechanisms, then the crosslink itself becomes your first point for degradation. For example, if you introduce an enzymatic sequence or a sequence that can be recognized by enzymes and cleaved, that makes the crosslink degradable, right? So again, to say that crosslinking is going to reduce the rate of degradation is broadly correct, but again, there are lots of subtleties when you make such statements, right? Now, when you look at amorphous versus semi-crystalline, an example given here is PCl, which is semi-crystalline, and PLGA, which is amorphous. Again, you have to relate it back to the ability of water to interact and penetrate the amorphous regions first, because of the disorder, whereas the crystalline lamellae are going to be much more resistant to water entrainment. It's going to take much harder for water to disrupt those secondary interactions and enter before they can cause hydrolysis, right? So, PLGA being amorphous is going to degrade much more quickly in a general sense as compared to something semi-crystalline like PCl, right? Another aspect to think about is stereochemistry of the backbone, right? Again, this can be tied again to regularity in the order, where something like PDLLA, which is largely amorphous, because of the lack of order, it's going to degrade faster compared to something like PLLA, which is more stereoregular, right? You can also have Tg of the polymer being important compared to body temperature, right? So, if you have lower Tg compared to say 37, then at 37 you have higher free volume leading to faster and higher water entrainment, right? You can also have hydrophobicity of backbone and side groups playing a very important role, just as we have seen, right? Poly-CPP, this is a polyanhydride which takes about 3 years to degrade. Simply by the introduction of the sebacic acid at 2080, now you have brought down the degradation time scale to a few weeks, right? And that can directly be linked to the hydrophobicity and relative hydrophobicity and hydrophilicity of the CPP and the sebacic acid. I would leave it to you as an exercise to look at the structures and convince yourselves that that is indeed true. So, you see, apart from the functionality in the backbone and the molar mass which we saw on the previous slide, you have at least some 2-3 more ways to tune and control degradation. One is the architecture, so this would be 3. The other is the amorphous and semi-crystalline. So, these two we can club them together because it has something to do with the regularity in order. And then you can have Tg, you can also have hydrophobicity or hydrophilicity. And shown here is just the ratio of glycolide to lactide in the PLGA copolymer, and what you are plotting on the y-axis is the half-life. So, essentially you are tracking degradation as a function of varying copolymer ratio. Now, when you have the pure polymers PGA and PLLA, the primary difference you see is the presence of the methyl side group here, which is absent here, right? So, PGA and PLA will have a certain degradation rate, but you see, once you start combining them in the copolymer PLGA in different ratios, the degradation rates consistently change, and that's largely because PLGA becomes amorphous. So, PLGA 8515, which would be somewhere here, is going to degrade slower than PLGA 5050, which would be somewhere here, right? So, depending on the type of application, you can pick and choose which PLGA you want based on how fast you want it to degrade and how long you want the material to persist. PLGA, we have seen, is an important polymer in commercial use in many applications. One particular application we have seen is in its use as a carrier for dexamethasone in the form of an injectable implant to treat diabetic retinopathy or to treat inflammation in diabetic retinopathy, right? Now, one other aspect to keep in mind when we make plots like these is when we say PGA has, we're giving actual numbers here, right? We're giving half-life in months. We do the same thing for PLA, we do the same thing for PLGA. One aspect to keep in mind is these are broad generalizations. Just as we have seen, the chemistry alone you cannot predict exactly what number you're going to have based on the chemistry alone. This is also a function of molar mass, this is also a function of processing. That is not something we have looked at so far in this lecture, right? If you process PLGA into matrices that have extremely high surface area, then you increase the propensity for water to interact with the material, making the degradation go even faster compared to casting films of PLGA, right? So, there's a lot of subtlety that needs to be kept in mind, but in a broad and general sense, from an academic perspective, one can still use such plots without making gross errors. The other aspect, again, as we have hinted, is hydrophilicity. Again, you see, this again has to do with access to water. As the hydrophilicity goes up this way, the percent degraded has increased. For the same time point, you get higher degradation and a faster degradation rate for a highly hydrophilic kind of a matrix compared to a hydrophobic matrix. And this again can be directly related to the ability of water to interact with the material and penetrate the material, right? So, those are some properties or some ways to think about how polymer properties affect degradation. Again, from a design perspective, from an engineering perspective, one can ask, how long do I want the material to perform, and what kind of degradation rates do I want? Depending on these kinds of questions and the answers to them, one can now engineer degradability. I think that's sort of the take-home message from these couple of slides. Now we will move on to looking for the rest of this lecture a couple of quick aspects related to specific degradation mechanisms. And the first one we'll look at is simply physical breakdown. And when we talk about physical breakdown, some aspects to keep in mind apart from the polymer. So, we talked about polymer properties that affect degradation. Here are some factors from the host environment. Again, you see, this is why we've kept insisting biocompatibility is not an intrinsic property of a material per se, because biocompatibility has to also do with the host and the material host interactions in a specific application. That's very important, right? So, these are some factors that have to be borne in mind from a host perspective. So, we've looked at the material perspective, now we're looking at the host perspective. Again, we've said water and ions, proteins, dissolved gases, pH, all of these can have an impact on the degradation. Of course, once you have cells infiltrating, cell secreted products, enzymes, and particularly you're talking about immune cells such as neutrophils, which are typically some of the first responders. Once protein has absorbed in a biomaterial, neutrophils are some of the first responders that will come and first inspect, right? In some sense, they are inspectors. They will come and inspect and see what kind of a material is this, right? And so, they will secrete enzymes that can be disruptive. So, we will look at some of those later in this course, but again, it's just to keep in mind that the host is also going to have an effect on the biomaterial, which can in turn lead to degradation. That's the point we're trying to make here. Of course, you can have special factors. If you have inflammation, chronic inflammation such as in osteoarthritis, then one question to ask is, if I'm using a polymer there, how long will it survive under those inflammatory conditions, which can exacerbate degradation, leading to unintended consequences, right? They are unintended only if you have not accounted for them. If you have carefully accounted for these factors, you will be able to predict the degradation rates and the performance of the material, even with these factors acting on the material itself. So, one aspect of or one mechanism through which biomaterials can degrade is simply physical breakdown or erosion, right. Then this is typically seen with mechanical forces acting on the implant. This could be wear and tear from prolonged use. It could also involve environmental stress cracking, examples of which we will look at in just a bit. And what you have to consider there is the debris that is formed and their effects. And these would typically be relevant in biomaterials and devices that are subject to mechanical loads, particularly repeated loading, right, such as ligament grafts, hip implants and so on. And now would also be a good time to recall a case study that we have seen very early in this course, the Vitek implant case study or Proplast, right. Now this was essentially material or a device made for the TMJ, temporomandibular joint, largely based on PTFE, right. And the issue there that led eventually to FDA recalling the implant was debris that came out due to repeated use, right. Again, remember, PTFE here, it's not the backbone that is degrading, right. It didn't lead to a decrease in molar mass because you're talking about carbon-carbon bonds which are extremely stable, right. Rather, what you're talking about is debris due to erosion arising from regular use, right, because of mechanical forces. So that's a good example to recall at this stage. And shown here, a more dramatic example is failure of these crowns, these polymer crowns on human teeth occurring from fatigue testing. Again, fatigue testing is something we have seen in some detail in lecture number 23. Now would be a good time to pause and recall, where we saw that devices and polymers subjected to repeated load-unload cycles, what you would call fatigue testing, are important for materials and devices that would eventually undergo those types of forces in the body at the site of implantation. So shown here is are pictures of failure of these crowns on human teeth occurring from fatigue testing. So here, this as per the study, they've looked at something like 900,000 cycles at 750 Newton force, right? Repeated loading and unloading cycles leading to eventual failure. So this would be what you would consider physical breakdown. This is a very important aspect of degradation because ultimately it's not just the backbone cleavage that's important. Things like these can cause catastrophic failure eventually, right, which would also be extremely detrimental to the performance of the biomaterial. Now, another aspect or another mechanism to think about for degradation is what you would call oxidative degradation, right. And we've just hinted at the fact that there can be these immune cells that can cause some kind of inflammation leading to some kind of localized degradation. And again, we are revisiting that and saying here that inflammatory responses arising from neutrophils, macrophages, phagocytosis is an important biological activity through which macrophages can sort of get rid of debris, right? And where they can't do that, particularly when the debris is very large, for example, then that leads to chronic issues, right? So the body has mechanisms to take care of minor faults, but when there are major design flaws, the body will be able to resort to some nasty mechanisms that will cause a lot of problem for the patient. So any active agents produced by pro-inflammatory cells such as neutrophils, macrophages, enzymes, those can all cause localized degradation, right? So for example, here we're looking at oxidative degradation. So you can have peroxides formed or free radicals formed that react with the covalent bonds leading to degradation, right? You can also have metal catalyzed oxidation, and some of these can be typically seen in polymer used in contact with metal wire in pacemaker lead. So, these are typically polyurethane plus silicone kind of insulations. So, we have seen in lecture 17 the use of polyurethanes in insulating the lead wires for pacemakers, artificial pacemakers. Because you have polymer in contact with metal, and because metal is present in a medium at 37 in the presence of water with salts, you can imagine that there could be mechanisms at work that lead to metal catalyzed oxidation leading to eventual degradation of the material, right. And shown here is another mechanism through which polyurethane can degrade, which we call environmental stress cracking, right. Again, apart from oxidative degradation, you can also have environmental stress cracking, where these cracks can lead to insulation breaches, right. So, if this is the polyurethane is protecting the wire, then such kinds of mechanisms where you have the environmental stress cracking occurring in polyurethanes leading to crazing, those kinds of failure mechanisms, then you can also have eventually insulation breach, which is going to lead to even more issues, because now you have direct access, or these cells etc. can have direct access to the metal inside, right. So, these are all problematic, right. And mechanisms through which these can occur include typically a free radical driven mechanism, and where a free radical site formation on the backbone leads to peroxide formation, where P is simply the polymer chain. In the presence of oxygen, you can have these peroxides that form, and peroxides again will attack. So, this is sort of the initiation step. The peroxide attacks the segment and abstracts the hydrogen, leading to propagation of the mechanism, because you see again more free radicals are generated here, and again termination can occur by recombination. So, these kinds of mechanisms are involved in what you would call oxidative degradation. And lastly, we are looking at chemical degradation, one form of which is hydrolytic degradation or hydrolysis, where again we have seen that chemical degradation is going to involve breakage or scission of covalent bonds, particularly in the backbone. It can also occur on side chains, but primarily we are looking at the backbone, and that is going to lead to a direct loss in properties, particularly molar mass, right. And as we have just seen, this depends on a lot of factors, particularly the susceptibility of the functional group in the backbone to degradation, and it can be exacerbated by factors such as the presence of enzymes, extreme pH conditions, etc. What is critical in all of these things, as we have repeatedly highlighted, is the access for the degrading factor or the species, either water or enzyme, if it is an enzymatically driven degradation mechanism, to the functional group for it to degrade, right. So, water penetration is a very, very important aspect for water to eventually cause hydrolysis, right. This is even important in drug release. So, if you make a matrix, right, and you have drugs loaded into the matrix, and if you implant this matrix, first thing that the water has to do is to penetrate the matrix and swell the polymer itself, right. Once the water has access to the drug gets solubilized and then the drug comes out, right. Unless that happens, the drug is not going to be effective at all, right. So, even for drug release, access to water becomes very important. Even for drug release from a polymeric matrix, access to water is a critical step for how fast or how slow the drug is going to degrade, which is why typically if you see bulk versus surface eroding kind of polymers, with surface eroding kind of materials, you will typically get zero order kind of, this is time, this is just a concentration of drug that is released, you will get zero order kind of release, where the drug that is released remains constant with time, right? Because the degradation happens layer by layer. Whereas in more bulk driven degradation, you will see an initial spike and then perhaps you will see it start to go down with time, right? So, these are some aspects to keep in mind, although they are not directly related to degradation. What we are trying to do here is to connect the concept of water penetration and access to different parts of the device and biomaterial to the performance of a biomaterial or a matrix in a particular application such as control release. Now, that being said, you can now have at least 3 versions of chemical degradation or 3 scission mechanisms. The first one, of course, is the backbone scission, where this is the polymer backbone. These are covalent bonds, and because of ester hydrolysis, for example, you can have these bonds breaking apart in the backbone. Another way for this to happen is for side chains, right? You can have side chains such as shown here that get cleaved, right? So, you can actually have materials where the design itself incorporates a therapeutic molecule, for example, on the side, such that the cleavage happens here and the therapeutic molecule can be released. That is one example of a side chain scission. You can also have cross-link scission, as we have seen. You can engineer, for example, these cross-links to be degradable, such that the backbone itself, while it does not really come apart, it is the cross-links that break, leading to the chains coming apart from each other. So, at least 3 different ways to think about scission mechanisms. One specific type of chemical degradation that becomes important is hydrolytic cleavage. And just shown here are 3 different aspects or 3 different functional groups for hydrolytic cleavage. On that spectrum, the timeline spectrum that we saw earlier, we are simply taking 3 of them: anhydride, ester and carbonate, and here we are showing how the hydrolytic cleavage can happen based on whether it is an anhydride, whether it is an ester, or whether it is a carbonate. So, these are reactions that should be fairly easy for you to understand if you keep in mind that they can all undergo hydrolysis, although to different rates. And shown on the right are examples of one each for each of the functional groups. Polyanhydride, again we have seen gliadel, which is used for glioblastoma treatment. Polyester, you can have lactosorb, which are used as meniscal staples, essentially some kind of a PLA, PGA kind of a polymer. You can also have polycarbonate, Maxon, which is a monofilament resorbable suture. Essentially, it is a copolymer of the glycolic acid and a trimethyl carbonate. Remember, here we are talking about carbonates. So, it is a copolymer of a glycolic acid and a trimethyl carbonate that is going to make it resorbable. So, just 3 commercial examples of polymers that incorporate the anhydride, ester and the carbonate functional groups. This is just for you to appreciate the fact that what we are looking at as concepts have direct implications for commercial use. I would encourage you to go look up the details of gliadel, lactosorb and Maxon from the manufacturer website, so that you get a good understanding of how the concepts we are looking at in lectures such as this actually translate to clinical applications. And before we move on, it makes sense for us to, again, because we talked about anhydrides and esters, it makes sense for us to talk about and quickly look at the distinction between surface erosion and bulk degradation, right? In surface erosion, what you're having is, with time, you're getting a drastic or a dramatic or a significant change in the shape and size of the material itself or the device itself. And that's because of the mechanism. Whereas in bulk degradation, again, depending on access, etc., how fast water gets in, how long it takes for the bonds to cleave, etc., you will not have this kind of a dramatic change in the overall shape and size. Rather, with time what you will get is bonds get cleaved throughout, and it's not just happening from the surface layer by layer. So this is the kind of degradation mechanism you will see. The other way to think about chemical degradation is for the agent to not be simply water but to be an enzyme, right? So that's the other aspect of chemical degradation, which could be enzyme mediated. And typically natural polymers that we have seen, many of which we have seen in this course in lecture number 19 and 20, many of them can undergo enzymatic degradation because enzymes show affinity for specific groups present on these natural polymers, right? One example we have seen is gelatin, like I just said on an earlier slide in this lecture, can be degraded by matrix metalloproteinases such as MMPs as shown here. But remember that this is not necessarily unique to natural polymers. Although it is predominantly seen in natural polymers, enzyme-mediated degradation can also be seen in synthetic polymers such as PLA and PCl through the action of proteinase K and lipases. And just as with water, the degradation rates can be determined based on the access of the enzyme to the active site. And particularly with enzyme, there are two more aspects that need to be kept in mind. One is the enzyme activity, which is also tied to temperature, and the sensitivity of the enzyme itself, right? One can, from a design perspective, one can take advantage of such kinds of enzyme-mediated degradation in biomaterial design. For example, if you are designing materials that can release drugs, for example, to control inflammation, now one can think about putting in MMP sensitive moieties along the chain, right? So this is the chain and this is the drug, right? So what's going to happen here is the drug is not going to be released when there is not sufficient MMP activity. Now you will see a high concentration of MMPs in inflammation. So when there is considerable inflammation, because there is going to be an excessive activity of MMPs, only under those conditions will these get cleaved and the drug gets released, right? So this would be one way to leverage what might seem like a disadvantage in your favor and try to design materials that can release drugs only under certain conditions through specific degradation or enzyme-mediated degradation mechanisms, right? And from a engineering perspective, you can also think about modeling this. Again, as we have seen, water penetration becomes important, and for the mechanism to be bulk versus otherwise, bulk degradation is typically seen if the rate of enzymatic bond cleavage is slower than the enzyme diffusion into the polymer, right? So we're talking about two time scales here. How long or how fast does the enzyme diffuse into the polymer, and how fast can the bond cleave, right? How long does that take? If the rate of enzyme entrainment into the material is faster, then you're going to get a bulk degradation kind of a mechanism, right? Whereas if the material starts to degrade right away as soon as in contact with the enzyme, then you will start to see an out to in kind of a degradation mechanism. And this bulk degradation can be modeled using Nicholas-Menten kinetics, which is a very, very famous standard equation for enzyme kinetics given by this equation, where V0 and Vmax are the initial and maximum degradation rates. This is the constant, and S is simply the degradable polymer concentration. Now in closing, now that we have looked at least the types of mechanisms that biomaterials can be degraded by, namely erosion, or then we've looked at chemical degradation, under which we've looked at hydrolytic and enzyme mediated. We've also looked at oxidative degradation. So these are some examples of mechanisms by which polymeric biomaterials can degrade. In closing, we want to ask this question: which is a question we've asked earlier, but we are revisiting that question now that we have a better understanding of biomaterial degradation. Is degradation desirable in biomaterial design? Answer depends on a lot of factors, and 3 important ways to think about whether degradation is important or not, or desirable or not in biomaterials are given here. One is the lifetime, the other is the location and function, third is the processing. All of these things are important apart from the polymer properties itself, right? Apart from functionality in the backbone, apart from side groups, apart from hydrophobicity, apart from crystallinity, apart from stereoregularity, all of which are intrinsic polymer properties. These the properties or the factors shown on this slide also have to be very carefully accounted for when you talk about degradation. First is lifetime. How long do you want the material to perform its job based on the function within the body, right? For example, if you want something like sutures, right, where if you want it to be resorbable, depending on the application, you're talking about days to weeks, perhaps to a couple of months, right? Because it's going to aid just temporary function, and until the time healing happens, thereafter the suture is no longer needed. Whereas if you're looking at bone fixation devices, right, you may still want degradation, but at a much slower rate, because you want it to provide support to the joint until the material can be integrated entirely with the bone. So it can go from months to several years if you don't want it to degrade, right? The other aspect to consider, of course, is location and function, where as we have repeatedly said, moisture, pH, enzyme, cell profiles, all of these have to be considered. And 2 examples are given here. For oral drug delivery, the polymer of choice has to be carefully accounted for, because the tablet or the coating is going to immediately come in contact with a gastric microenvironment, right? Or if you are using it in an inflammatory microenvironment, there are other processes that need to be accounted for, such as oxidative degradation. Of course, degradation is also going to lead to a loss in mechanical properties, and this is something we have looked at or hinted at when we looked at lecture 24, and we saw that processing can lead to porosity, can result in degradation, can lead to adverse effects on mechanical properties. And here the point to be kept in mind is the degradation time needs to be carefully matched against the biological repair time frame for any function. An example is load bearing function. So, for example, if you are plotting time versus degradation, or you plot it this way, loss in mechanical properties with time, right? These would be what you would call mechanical properties of the material are going down with time. Now you can have an increase in tissue healing, right? So at some point, this is tissue growth. So at some point, you see the tissue is going to take over, the new tissue is going to take over the properties, which means that the degradation rates for that material or device have to be carefully balanced against the tissue growth rate, such that the mechanical properties don't suddenly change over time. So gradual decrease in mechanical properties has to be balanced against the slow tissue growth rate. Of course, processing is going to have a very important role, because processing, which we will look at in a dedicated set of lectures very shortly, is going to change the shape, size and surface area of the biomaterial, and all of that can have an impact on how quickly water can access, how fast it can degrade and so on. And lastly, one other aspect to keep in mind is sterilization is often done for these materials before they can be implanted. It's almost always done. And what is the effect of sterilization, right? If we're using radiation, for example, right, gamma radiation to sterilize, does that induce any processes within the biomaterial that can cause it to degrade faster? All of these factors have to be kept very carefully in mind. So in closing, I would like you to pause and reflect on this very important idea. if you want to use polymeric biomaterials for as sutures or for oral drug delivery or for bladder replacement or as bone fixatives, right? What kind of polymers would you want to look at? What kind of properties would those polymers require to have? And lifetime, location, function and processing. These three factors, how are they going to be contextualized for each of these things, right? So, if you have been able to understand the structure property relationships that we have seen throughout this course, as well as the degradation aspects of polymeric biomaterials that we have looked at in this particular lecture, you should be able to quite easily come up with some broad ideas for the design of materials to be used as sutures for oral drug delivery, bladder replacement and bone fixatives. With that, we will end this lecture.